Just like with the mitochondria, you need to know the basic parts of the chloroplast. And the chloroplast is the main way that living organisms harness solar energy for life, which is very important. The inner and outer membranes are a lot less involved than they were in mitochondria, but I still need a lot of surface area for chemical reactions. And the chloroplast meets this need with the thylakoids. The thylakoids are these discs that are arranged in stacks called granum and interconnected with each other with this membrane layer called the stroma lamellae. And that's really just like a fluid exchange and material exchange for the granum. The fluid inside the chloroplast is called the stroma, and the fluid inside the thylakoid is called the lumen. And that's going to give you place locations for some of the stuff later, so make sure you remember which one's which. This organelle did come about through endosymbiosis, so it has its own DNA, its own ribosomes, it makes its own proteins, and don't forget that. To first understand photosynthesis, you must first understand light. So we know that light is in different wavelengths between red and purple and violet. But if you look at the actual absorption spectrum of plants, which is the colors that they are absorbing versus reflecting, we see this big drop in the middle. And we know that it's in the green section. Why does that make sense? Well, plants are green. That means that they are reflecting green light, not absorbing it. Now, you know chlorophyll is the green pigment in plants, but you might not know that there's actually more than one kind of chlorophyll, at least 5 that I can think of. But there's 2 very common ones. Chlorophyll A is the blue-green, chlorophyll B is the olive-green, and together they're going to function as they allow light to be absorbed and transfer that energy to the reaction centers of the photosystems in photosynthesis. So they're very important for photosynthesis. If you look at the diagram below, however, you'll notice that all the pigments together work to form an action spectrum. And the action spectrum is widely different. So it's a sum of all of the different pigments in plants, because there's more than just chlorophyll. You also have carotenoids, like carotene, which is the orange in carrots, or the anthocyanins and purples in flowers and so forth. These other pigments, carotenoids and anthocyanins, function for photoprotection. They're called accessory pigments. Basically, if there's too much light, you need to be able to absorb and reflect different light than just photosynthesis needs to protect the plant. Sometimes it's called the xanthophyll cycle, but regardless, all the other pigments help work preventing too much energy from being absorbed. So, plants are producers. They are autotrophs. They're able to take solar energy and turn it into simple sugars. Glucose can be used by the cells for energy to make ATP during cell respiration. The sugars produced by photosynthesis also provide carbon skeletons that can interact with elements such as nitrogen and sulfur and phosphorus to make other organic molecules, amino acids, lipids, or nucleic acids. So the formula of photosynthesis provided is for the production of glucose, but it is not the only way that photosynthesis can occur. It's just the most common, and it's the one you need to remember. As a side note, it is just the cell respiration formula flipped around, so hopefully easy to remember. Now, where does photosynthesis occur? We know it occurs in the chloroplasts, but the chloroplasts are mainly in the mesophyll. So if you take a leaf, it's that spongy tissues, the spongy cells that fill up that leaf, and each leaf has hundreds of mesophills, if not thousands, and each mesophyll has between 30 and 40 chloroplasts, depending on the species. If you take photosynthesis, you can really just split it in half right down the middle. So on the left, you have the light reactions, which are the energy building reactions. The light dependent reactions is the more current name. And on the right, you have the Calvin cycle, or the light independent reactions, or the sugar building reactions. So the left makes energy, the right makes sugar. So that's really it. So solar energy comes in, that energy is harnessed to produce ATP and NADPH. That NADPH and ATP is used to power the Calvin cycle, which produces the sugars. And the waste from the Calvin cycle are reused in the light reactions. The light-dependent reactions occur in the thylakoid membrane because I need membranes to form a proton gradient. That proton gradient is going to build inside the lumen to power photophosphorylation, which is a little bit different than we've seen before, to allow ATP synthase to produce ATP. But that's not the only thing that light-dependent reactions do. So when light comes in, it strikes the chlorophyll. The chlorophyll shoots an electron off, and that process falls down electronegativity through these different proteins until it hits photosystem 1. Different protons are pumped in using that energy to build that gradient for ATP synthase. Once that electron gets to photosystem 1, more light comes in, and that shoots the electron up again to where it falls to capture more energy to power a second enzyme, NADP plus reductase, which produces my electron carrier NADPH. This is the only electron carrier that plants need. Now, this process is very similar to the electron transport chain in mitochondria. So if you look at that, you can see that the structures are very similar. It's all just different directions and names for where they're occurring, so make sure you understand the differences. The light-independent reactions, or the Calvin cycle, do not require solar energy. So it's not called the dark reactions. That's a misnomer because it happens in the light or the dark. It just does not require energy from light. Therefore, it is light independent. This occurs in the stroma, not in the thylakoid at all, in the fluid inside the chloroplast. And there are 3 major stages, which we'll look at in a little bit. But the big thing here is that you take in CO2, you use the energy produced by the energy-building reactions, or the light-dependent reactions, here, here, and here, to produce sugars. For every 3 CO2 that you bring in, you produce 1 G3P, which is a base sugar to build lots of other things. So if I have 2 G3Ps, I can have 1 6-carbon glucose. For me, it's simpler to think of the Calvin cycle as 3 separate steps. You have the carbon fixation step, which you take a 5-carbon RuBP, combine it with a 1-carbon sugar to produce 3-carbon PGAs. Now, it'd be nice if we could stop there, but PGAs are not stable and they're not going to be good for long-term sugar usage. So you then go on to step 2. The PGAs are rearranged by applying ATP and NADPH to produce the stable G3P. Now, so ATP is going to provide the energy I need for this, but the NADPH is going to be what's giving it its reducing power, which is very important for producing things, because if you don't remember that, go back and listen to oxidation and reduction again from the previous sections. So that's going to produce 6 3-carbon G3Ps. For every 3 CO2s that came in, 1 G3P can be released, and the other 5 will be rearranged using a little bit more energy back into the RuBP I need to keep the whole cycle going. So it's a constant cycle that produces sugar by using the energy of ATP and the reducing power of NADPH. Calvin cycle is all well and good, but how in the world do you take a gaseous CO2 and turn it into a solid sugar? Well, that first step of the Calvin cycle, where the RuBP is combined with the CO2, uses a special enzyme called RuBisCO. That stands for ribulose bisphosphate carboxylase oxygenase, but you don't have to remember that. Just remember RuBisCO. It's probably the most important plant enzyme, and it's probably the most common plant enzyme, because it's about 50% of all the proteins in all the plant leaves pretty much on the planet. It takes that CO2 and it uses that reducing power of RuBP to reduce it into that solid. Because if I change the structure, I change the function, so I changed a gas to a solid. Very handy. That process is endergonic. It takes energy, and that's why I need ATP, and that's why if you actually study like thermal images of plants, they're going to be a little colder because it's going to be taking energy in. Now, RuBisCO is old. Okay, it's been here for about 450 million years as far as we can tell from a big spike in oxygen at that time that we can see in the rocks. So it can get really confused because it evolved before oxygen was common. So like we said, RuBisCO is old and it can get confused. And the reason it gets confused is because it gets hot. So Plants have these openings called stoma. And when it gets hot, plants close those stoma. So oxygen is going to build up because of the light reactions, and CO2 is going to go down because of the Calvin cycle. And that means that RuBisCO is going to be exposed to a lot more oxygen. So RuBisCO can get confused. It cannot tell O2 apart from CO2 because it evolved at a point in life where CO2 was common and O2 was rare. So that means that instead of reducing the RuBP like it should, it's going to oxidize it. It's going to break it down, and no ATP, no sugars will be produced. Now, this is a big problem for plants. If you could reduce this, they would become much more efficient. And that's actually going to be a strong selective pressure. It's going to make evolution much more likely because plants that can do this will survive better, have more offspring, and pass those traits on to their offspring. Diverse groups of plants have evolved different systems for coping with the problem of photorespiration. So plants that use traditional photosynthesis we call C3 plants because they make 3-carbon sugars. There are 2 other big classes of plants that have found ways around the photorespiration problem. The C4 plants physically separate the fixation of carbon from the Calvin cycle by doing it in a different cell. Okay, so they're going to have altered leaf structures. It's an anatomical, physical separation. Corn, sugarcane, things like that do this. Basically, if you have it separated, it doesn't matter if oxygen's coming in, the CO2 will be taken somewhere else. Now, it does this by using an enzyme called PEP carboxylase, and that is going to take CO2, it's going to store it as an organic acid, and transport that to this other cell. Now, canned plants are a little different. Instead of transporting it to another cell, they do it at a different time of the day. These plants tend to have evolved in like desert environments or tropical environments, depending on if it's high humidity. Pineapple and cacti and succulents. Basically, you take that CO2 in at night when it's safe to have your stomates open, and that is going to allow you to take in lots of CO2. You're going to take that CO2, you're going to fix it into organic acid using PEP carboxylase, and you're going to hold on to it until daytime. In daytime, you're going to close those stomates to keep that water in, and that is going to have you have a buildup of CO2 that you can use. So if you look at this overall, C3 plants take in the CO2 and go through the cycle like normal. So this is my C3. C4 plants are going to make a 4-carbon organic acid and transport that organic acid into another cell, whereas my CAM plants are going to fix the acid at night and use it during the day. If you actually look at how photosynthesis and cell respiration are connected, it's quite interesting. So you take in sunlight. That sunlight is taken in by an autotroph. That autotroph does photosynthesis to produce organic molecules and oxygen. That material is digested by heterotrophs in cell respiration to get energy. The waste products from that digestion, CO2 and H2O, are the incoming products to photosynthesis. So these separate linear processes are connected. They are coupled to keep the energy flow alive and keep other organisms functioning on this planet.